It looks like it's 4 o'clock. Usually we have participants dropping in right around 4, so I see we still have some folks joining us, but I think we'll get started. Welcome everybody to today's Hutch at Home series. I'm Jeannie Chowning, Senior Director of Science Education at Fred Hutch, and we're delighted to be able to bring some of Fred Hutch science to you in this way. And especially want to acknowledge the difficult time that we are in right now, and I appreciate you making space today to come join us, and I hope that the teachers here and the students here all taking good care of themselves, and I encourage you to look at SEP's statement, which is posted on our Facebook page and our social media, about our concern for the events that are happening and our solidarity with especially our Black colleagues who are bearing the brunt of all of this upheaval. So I am really excited today for our speaker, Julie Overbaugh, who will be talking about the power of international collaborations, and specifically an example from studies of HIV infection in infants. And I think collaborations and partnerships is a great theme for today. Okay, I would like to, please mute yourself if you're not talking. A quick reminder, looks like I got halfway through a word there, but it's especially if you are on a phone. I know it doesn't automatically mute you if you come in on a phone, so check that please. And then feel free to add your questions into the chat and complete our feedback form at the end of the talk. Okay, so Dr. Julie Overbaugh is our featured speaker today, and she's a professor in both the human biology divisions and the public health sciences divisions at Fred Hutch. And she's also the endowed chair for graduate education at Fred Hutch. I have been working with her, particularly in her role as director of the Office of Education and Training, and I appreciate the passion and commitment and advocacy that she's brought for science education through that role. And she's also the associate director of the Cancer Research Career Enhancement, part of the Cancer Consortium with Fred Hutch, UW, Seattle Children's, and Seattle Cancer Care Alliance. I also want to acknowledge that this year she was elected to the American Academy of Arts and Sciences, which is a big honor, and we celebrate her for that as well. So with that, I'm going to turn my screen over to Dr. Overbaugh.
Thanks, Jeannie. I'm going to try sharing my screen. I have to admit that I'm computer impaired, but I'll do my best. Desktop 1. Let's see. Does not want to do that. Let's see. Okay.
We can also, I can run it for you and you can say next slide. That's another option.
If that works, that might be better because it's asking me for privacy updates.
Okay.
And in fact, you don't all want to go through privacy.
Yeah, well, no worries. Let's try this here. And if someone would be willing to just verify for me in the chat that it all looks good, that would be great. Okay.
And then for questions, Jeannie, are you going to see them? Should I be looking? What would be the best thing?
I can look for you and let you know when there's a question that's emerging. So I'll keep an eye on the chat for you.
Okay.
And so you just let me know when you want the next slide.
All right, thank you.
Okay, can you see it?
I cannot. Can anyone else?
Let me try that again. Okay, can we try again? Sorry for the technical difficulties, my friends. Okay.
There we are. Excellent. I guess, yeah, okay, that is great. Thank you. So I want to today just tell you a little bit about some of the work we're doing on HIV antibody responses in infants, but I want to give you kind of a context for the way my lab thinks about the problems that we approach. You know, a lot of people study model systems, they study fruit flies, or they study cells in culture. And, you know, really one of the central things that we think about is when we address our scientific questions, we want this to be as much as possible in biologically relevant to infection in humans. And we study HIV transmission and disease, but we've also become interested in emerging viruses such as Zika and, of course, the coronavirus, SARS-2 coronavirus. So we try to understand the biology of these viruses and disease in human populations, and we do that by leveraging collaborations that we've had in Kenya for over 25 years, and that's something I'm really going to try to emphasize as some of the collaborative work that we do. So next slide. I'm going to talk about antibody responses against HIV, and we study that in the context of mother-to-child transmission of HIV, and also in the setting of people who get reinfected or superinfected. So they have HIV from one source partner and they get infected again. And that's an interesting setting because it means the immunity failed. And that's also relevant to a lot of the things we're thinking about with the coronavirus now, because what we really need to know is whether the immunity that's elicited to that virus actually protects against infection. Next slide. So I'm gonna just talk a little bit about me and my origins. It sounds like that's something that people like to kind of understand about where scientists came from, and my background is a little atypical, I think. And then how I got started in the international research arena, and that was in 1992. And then we'll talk about protective antibody responses, and you know, I'll start by trying to explain why we care about that, and maybe that's some of that's kind of obvious now as you're reading about coronavirus, but you know, you could also be thinking, what are the challenges? Why haven't we figured this out for HIV? And then I'll talk about what we're learning from infants. But at the end, I want to come back and just talk about this theme of international collaborations and really what they bring beyond the science, just to sort of wrap the whole thing around. And yes, feel free to ask questions and interrupt through the chat box. Okay. So a bit about me, the next slide. So I grew up in a working class town where sports were more important than academics, and had a lot of different jobs. I was a waitress, I worked at the 7-Eleven, McDonald's, but one of my really most memorable one was the couple times I worked at the pretzel factory. The town I grew up in is known for its pretzels and its thoroughbred horses, and they were sort of foundational parts of my growing up. I worked underage in the pretzel factory, as people did in those days. I know a number of you are teachers, and I'll show you this next photo because I probably was not one of the favorites of the teachers because I was a little bit nonconformist. And you can see that in this picture here with my cousins and my sister for the May procession, where they all wore the appropriate dresses, and I showed up in my brown dress for that particular occasion. So that was kind of I was a kid who, you know, sort of liked to go my own way a little bit, and especially in that small town. The other thing that was really sort of central to me is sports. And I played basketball for the University of Connecticut, and I actually picked University of Connecticut for the basketball team and the tennis team, which I played on. And I never actually even thought to look and see if it's a good chemistry department, which was my major at the time. So I grew up also more focused on sports than academics. Next slide. So I've gotten a little too old for basketball, but I still love to be outdoors, and I find that actually being outdoors and being active and having outside interest is pretty important to staying grounded in science and being open and creative. And I've actually done some writing about work-life balance in science because I think it's important to have, and that really what as scientists we need to do is be thinking about ideas and be creative, and that doesn't necessarily come sitting at a computer and staring at it all day. So I still stay very active. Okay, next slide. So I'm going to talk about sort of where I began, and one of the reasons I wanted to bring this up is really the parallels that I see from when I was studying retroviruses, which HIV is a retrovirus, and where we are now. So this was 1985, and there I am working at the lab bench. And at the time, we had just gotten a first antibody test for HIV. So this kind of sounds like a parallel to where we are with coronavirus now. AIDS had been reported in 51 countries, so it was pandemic by that point. Any of you who are old enough will remember that one of the sort of aha moments that this is a real thing was when Rock Hudson died of AIDS. Mm-hmm. And about that time, there were about 12,000 deaths in the US. So this kind of reminds you of early spring 2020 with the SARS coronavirus 2, which is the virus that causes COVID-19. So I find that this really is reminiscent of the time when I started studying viruses. What we're seeing now reminds me a lot of what we were seeing with HIV in those days. And if you look at the outcome of where we are now, all these years later, there's been 75 million people who've been infected, and there's been 32 million people who died of AIDS. So this was a problem that was just starting and had a long way to go. And in fact, it took many years. It took 20 years before we even started to put a dent in mortality from HIV. It was only in 2005 that things started HIV, so that took us 20 years, and of course those therapies are quite important now. But you know, the point is that there are many people living with HIV still, and the majority of those are in Africa. So while we've made a big dent in the epidemic in the US and in developed countries, we still have a major epidemic and pandemic in Africa. Okay, next slide. So how did I get started in international research? Next slide, please. So when I was just starting my lab in the early 90s, I met these two women, Joan Price, who was at the University of Washington, who was an epidemiologist and clinician, who had gone to Kenya to work and was one of the first people to describe HIV in that part of Africa. And she had partnered with Ruth Ndwati, who's a professor at the University of Nairobi. And at the time, they were very interested in this question of whether or not breastfeeding was responsible for transmission from mother to infant of HIV. And at that time, it was actually recommended that women who were HIV positive continue to breastfeed because it wasn't known what the risk of transmission was. And we knew that in developing countries with not access to good water supplies, that there was also a lot of mortality and morbidity associated with formula feeding. So the risk of breastfeeding was unclear. We knew it could transmit HIV from case reports, but we didn't really know how frequently that happened. So they did what's considered the state of the art in understanding these kind of problems, a randomized clinical trial. where they compared HIV infection between breastfed and formula-fed groups to determine the frequency and timing of breast milk transmission from mother to infant. And of course, the formula feeding group was identified as people who did have access to safe water supplies, and they were provided with formula. But at the time, there were no treatments for HIV. So this was treatment naive times in the HIV world in the early 90s. And they remarkably followed 425 women and infants through 2 years of life in this study. And I became involved with these great women because I was a virologist and there were no good assays for them to detect HIV in infants at the time. So they couldn't look at their endpoint without a good assay to detect HIV. So I was working on other viruses, but I said, sure, you know, this looks like an important problem. And I joined the team to provide viral assays and develop those assays, not knowing that this would sort of be the launching point for me in terms of the direction of my research. Okay, next slide. So this shows you the outcome of the Nairobi breastfeeding trial. And in the green is the breastfeeding arm, and in the blue is the formula feeding arm. And we're comparing percent of infants infected over the life of the infant through 24 months of age. And I think you can see immediately, next slide, that at 24 months post-infection, there's almost double the number of infected infants in the breastfeeding arm. So this told us that breastfeeding actually was responsible for about half of the infections among women who breastfed. And next slide, what you can also see is that even as early as 6 weeks of life of the infant, there was a significant difference in the number of infants who were infected in the breastfeeding arm. So this was important because at the time, you know, we didn't know if the risk was early for transmission or over time for breastfeeding transmission. And this did tell the field and people who were thinking about trying to prevent transmission through breastfeeding that we really needed to intervene in that early window within the first weeks of life. And this was a published study that Ruth led, a really important study at the time that really clarified a lot of these important public health issues for mothers and infants. Next slide. So there were a lot of other things that came from that trial that were really important as well, that were led by colleagues of mine. And these are just two that I picked out that I think really are kind of a sobering results. And one is a study by Dorothy Mbori-Nagacha, who was a pediatrician and also a professor at the University of Nairobi who worked on this project. And she simply asked the question, if an infant gets infected early in life, within the first 6 weeks of age, compared to an infant that got infected later, what's the mortality in those two groups? And if you look at the bar graph, you can see that in the infants that got infected before 6 weeks of life, before there was treatment, 60% of them died by 2 years of age. It just remarkable. Whereas the infants that got infected later in life did much better. And we kind of came to understand part of the reason for that, and that is that infants had higher viral loads, and that's shown in the right graph. Comparing the same group of infants, early and late infected infants, you can see that the viral levels are much higher in those that got infected early. So for some reason, they can't control their infection if they're infected really early in life. Next slide. And I just want to step back a minute and say why viral load is important. And again, it's something that we need to understand for the coronavirus as well. But what we understand for HIV is that if people have a higher viral load, they progress more rapidly to disease and they are more infectious to a partner. And that's true for mother-infant transmission too. So the more virus, the more rapid the disease, the more infectious for HIV. And this is something we've come to understand by studying viral load in relation to these outcomes. It's something we don't yet understand for the coronavirus. Okay, next slide. Sorry, next one. Forgot about that animation. So, you know, here I was, this kid who grew up in a small town in Pennsylvania. I actually didn't get on my first plane until I was in my 20s. But I did then get on a plane and go to Africa for the first time in 1993 after starting to be part of this collaboration. And actually, we went to partly set up some work for the study I just talked about, but we also set up another study at the Mombasa Hospital shown here, and a sexually transmitted disease clinic in 1993 to study high-risk women. And we've actually followed this cohort of women for over 25 years now. That's me on my first safari with the elephant in the background. And over the years, we've built an impressive lab staff. We've gotten lab space and gotten that set up. And actually, this is relevant to the new coronavirus pandemic as well, because our staff now is poised to help with the response to this new virus. Next slide. Okay, so let me talk about protective antibody responses. And again, I'm very open to questions, and especially about the science part might generate a few more questions. If it's not clear, just interrupt. Okay, next slide. So I think you're probably all hearing about antibodies a lot, again, with the coronavirus efforts, that this is really what people are banking on to be protective. And even today, there was a report on one of the vaccines that's being tested saying, you know, it makes antibodies. So this is really, you know, for most vaccine efforts, this is a major focus, and it has been for HIV vaccines for a long time. And, you know, part of that is simply because most vaccines are thought to work through antibody-mediated protection. But in the case of HIV, there's also been a plethora of studies showing that if you put antibodies in non-human primate model systems, you can protect them from an HIV-like virus. So there's proof of concept in an experimental system. Next slide. Let me step back and just talk about what neutralizing antibodies are after giving you that sort of reason why they're of particular interest. So this schematic just shows you the virus on the top and the target cell for infection, which for HIV is a T cell usually. And what generally happens in an infection is the envelope protein, which is shown in that kind of blue color, engages the receptors on the cell and enters the host cell and reproduces itself, and that's how the virus disseminates. But antibodies that can bind to that envelope protein and block entry are called neutralizing antibodies. So they neutralize the virus, they don't let the virus enter the cell. So for HIV, that protein is called the ENV or envelope protein. For the SARS coronavirus 2, that is called the spike protein. Oops, we just went back. There we go. So again, you'll hear a lot about the spike protein and neutralizing antibodies to the spike protein. Same principle there as what we've been trying to do with HIV, which is find an antibody that binds the protein, blocks it from getting into the cell. Okay, next slide. So, you know, I emphasized that this pandemic was starting in the early 80s. And here we are in 2020 without a vaccine. And on that last slide, you know, you could look at that and say, that looks like that shouldn't be such a hard problem. But really, it is a hard problem for HIV. And that is because, well, for many reasons, but one being that HIV mutates at a high rate. So this schematic just shows you that. Again, the virus is shown on the top. In the absence of antibodies that neutralize it, it will bind to those receptors and get in the cell and reproduce itself. It's called a retrovirus because it goes the opposite direction of the flow of genetic information in your cell. It goes from RNA to DNA. And in that process, it often makes a mutation so that the virus that comes out of the cell carries that mutation, which you can see with the red flag that indicates the effect of a mutation. So this is a really error-prone virus, and that is true of a lot of RNA viruses, and coronaviruses are also RNA viruses. Next slide. But just to give you a sense of the magnitude of this variation, I show here at the top two panels are comparing two different people and looking at the sequences between any two of you over the course of a 10 kilobase region of the genome. And if you did that, you would see about 0.1% change, and that's indicated with the red lines. So about 1 in 1,000 bases are different between you and the next person you're not related to. If you compare HIV genomes across the same region between two people, they can differ by as much as 30%. So this is a remarkable amount of genetic variation just within HIV type 1. So the next slide shows you this in a different way. So one way that we categorize these viruses is we create what are called phylogenetic trees. And those trees group viruses by how closely related their sequences are. So in this sort of circular tree, you see the red sequences are more related to each other, and they're actually considered a different subtype or clade of HIV-1. And then you have the blue sequences at the other side, and they're a different clade or subtype. And those red and blue sequences will differ by about 30%. So I show you on this phylogenetic tree that that means there's all different viruses, HIV viruses, that a neutralizing antibody has to be able to recognize in order to neutralize them. So it's not going to be effective if it only neutralizes the green virus, because anybody's exposed to the other viruses won't be protected. And so this is the challenge of eliciting neutralizing antibodies to HIV. But it has been exciting news in the last decade, because we've identified antibodies that are capable of neutralizing these diverse viruses. And these are called broadly neutralizing antibodies, or BNAbs. And that next slide. So these BNAbs I just illustrated here, that's an example of an antibody that recognizes all these different viruses and can neutralize it. And this is really the holy grail of some of the HIV vaccine efforts. If we could elicit these BNAbs and at high enough levels, we should be able to protect against diverse strains based on the idea that they protect in monkeys, and a lot of vaccines work this way. Okay, next slide. So the other problem, and I'm going to dig a little deeper here, so again, feel free to raise your hand if it's not making sense. You could make a broadly neutralizing antibody, but if it wasn't work. So
Julie, you cut out for just a minute there, so could you repeat your last sentence, please?
Yes. So you could have a neutralizing antibody that's broad and a BNAb, but it also has to be potent. It has to be able to deal with all the virus that it encounters. And the problem with antibodies is they don't actually start out being very potent. They actually have to mutate to become potent. So I'm going to explain that with this schematic. So B cells produce antibodies, and we have what's called a repertoire of naive B cells. Those are cells that have not seen the protein or the antigen they're going to respond to yet, but they're ready. And the way our immune system works is we have a lot of those B cells ready to respond to different infections, different antigens, because we don't know what's coming. So we didn't know if we're going to be exposed to Zika, dengue virus, HIV, coronavirus. So we have this repertoire of cells that's kind of ready, but they're not optimal. But when they do bind to their virus and get stimulated, they will start to go through a process that's called somatic hypermutation that allows them to get better and better at binding their virus or antigen. So this makes the antibodies that are secreted from the B cells more potent. But what's troubling with the discoveries of HIV BNAbs, next slide, is that when these have been studied in cases of infection in adults, some studies have shown it takes 5 to 10 years to get these BNAbs. So this process of generating this BNAb and this perfect B cell that makes the right antibody would take too long for a vaccine. And it's thought to be happening partly because the virus mutates, the antibody mutates, the virus mutates to get away from it. There's what's called an arms race between them that plays out over years. So we do know that we can make BNAbs, that people who are infected with HIV do, in some cases, not many, but in some cases make these BNAbs. But we don't think it's very practical to mimic this kind of situation with a vaccine. We don't have time to immunize people year after year to get there. Okay, next slide. So let me just stop and see if there's anybody who puts a comment in the chat about questions on that before I talk about the infant study.
Only comment so far is being impressed with your basketball record. Somebody looked up and noticed your stats and shared them and was quite impressed. So no other questions so far, but please feel free to add your questions into the chat.
And full disclosure, I was not the star of the team. I was just too scrawny and not scrappy enough, but I loved it. Anyway, so what have we learned from infants about making good antibodies? Next slide. So we kind of stumbled on the idea that infants may hold answers to how to make these broad and potent HIV neutralizing antibodies that I said were sort of the goal of HIV vaccine efforts. And this was actually a study that was led by a student in my lab, Leslie Gu, who's now become a faculty member at the Hutch, just recently joined. And one of the things that enabled us to do a study of infant antibodies was the Nairobi breastfeeding trial that I mentioned, because we had samples from that trial available to study. It was before there was treatment. And without that access and collaboration of the investigators in that trial, we would never be able to do this study. And in fact, you could not do this study now because infants are successfully treated for HIV infection. So what Leslie did is she identified infants in the trial who became infected and who had follow-up of about 2 years of life, because we knew, as I said, from adults that it takes quite a while to make these BNAbs. So she then looked at those 28 infants and screened their plasma for the ability to neutralize these different diverse viruses from different subtypes and clades that I had shown you on that phylogenetic tree. Next slide. And what she found was remarkable in that 70% of the infants she screened had broadly neutralizing antibody responses in their plasma. Now, this is the plasma response. It's not a BNAb, a monoclonal antibody like I was talking about before that neutralizes diverse viruses. But it said that somewhere in the infant, there was the capacity to generate broadly neutralizing responses. And what was also remarkable about that is that the 70% that she identified was in contrast to what had been reported in adults, where only about 20% of adults actually generate these highly desirable responses. So, you know, what we learned from that is that infants develop broadly neutralizing responses more commonly and more rapidly than adults. So this was exciting, and we thought that they had something to tell us about how to make these responses. So next slide. A very brave graduate student in my lab, Cassie Simonich, decided to go back to those infants. We had been storing samples in our lab since the 90s and pull out one of those blood samples and try to identify the monoclonal antibodies that were present in that infant. And to do that, first of all, we were excited that the sample was viable and we could do the experiment. But basically, Cassie had to sort through about 100,000 B cells to find 11 that recognized HIV. So this was a tour de force, but the results were interesting. Next slide. Because she did identify one what we call BNAb, and that's BF520.1 shown here with the red color. But if you think back to what I showed before about a true BNAb, I talked about those as having the capacity to neutralize all the different viruses. So this was broadly neutralizing. It was a BNAb, but it didn't neutralize every virus we tested. Rather, some of the other antibodies, next slide, that she identified were responsible for that breadth. So this was really interesting because it told us in this infant, it did in fact make a broadly neutralizing antibody. And this was at 1 year post-infection when we sampled we took the sample, so much faster. But the response to get the full breadth was polyclonal. And this is exciting to me because I worry that if all we try to do is develop a really, really broad single antibody, that the virus will escape. Whereas if we have this polyclonal response, it just creates a much bigger puzzle for the antibody repertoire to not permit the virus to escape so easily. Okay, next slide. So one of the things we really have wanted to understand is how did that infant make that response? Because the way a lot of people in the HIV field think is, and I think this is a valid approach and one that we are following, is that if we can understand how it was done once, we could go back and try to recreate that experimentally to drive the same kind of response with a vaccine. So we decided to go back and try to understand where this antibody came from, what was the naive antibody, naive B cell that started this process, and how did it get there? And so to do that, we just sequenced the entire repertoire of antibodies at several time points in this infant from before we isolated the antibody. So again, this is really only possible because of the Nairobi breastfeeding trial, where sequential samples were collected and stored from these infants. Next slide. We then teamed up with some mathy types at the Hutch who do computational biology. And what they did was go back through all those sequences and ask, what's the likely path from naive to mature antibody for this particular antibody lineage, is what we call it. So how did we make BF520.1? So they made their best estimate of how this happened based on all the sequences that we generated. And this was led by Laura Dopker in my lab with Eric Matson's group, who's just a phenomenal computational person at the Hutch. Next slide. So I'm going to walk you through the results, and I'll tell you that first of all, what we did is when Eric's group told us how they thought this antibody evolved, we went back and made those antibodies, we generated them, and we tested their ability to neutralize HIV from diverse clades. So if you look at the bottom panel, all you have to pay attention to is that gray means they didn't neutralize, blue means they did, and the darker the blue, the more potent the neutralization. And there's just different viruses listed in the table. But I'll also show you a cartoon version. So one of the things that was interesting is what we predicted to be the naive antibody did not neutralize the virus, but it had just enough ability to bind HIV. So we think it was able to initiate this process of stimulating the B cell and starting somatic hypermutation. Next slide. When we added just a few mutations that we thought happened as this antibody evolved to become the mature antibody, we could see a little bit of neutralization. You see there's a little blue box in the panel below. And in the schematic, you can see there's very narrow neutralization of just one virus. Next slide. As we kept adding mutations, we got more breadth and more breadth. Next slide. And more breadth. And next slide. And then we saw additional mutations made the antibody potent. Sorry, we can go back one. So we were able to reconstruct what we think was the slow but steady process of this antibody lineage to make this BNAb in an infant within a year after their infection. Okay, next slide. So this just summarizes what I told you that looks to be different from adults and infants in terms of their neutralizing antibody development to HIV. In adults, it looks like they make these broadly neutralizing responses mostly as a single monoclonal response. One antibody just gets better and better and better and more potent. And that explains a lot of what we see in adults. It doesn't explain it all, but that's mostly what we've seen in adults. And it's just interesting and fascinating to me that infants seem to approach this differently. They generate BNAbs. They're not as broad, but they also make a polyclonal response where other antibodies seem to chip in and collaborate with the BNAb to really give it the full breadth and potency of the response. And it's also exciting to me that, next slide, the process seems to be accelerated in infants. So this process of generating the right antibody through mutation takes a long time in adults, and somehow infants seem to have accelerated this process. In fact, in our studies that I just showed you, we found that it took only 2% of that mutation to get the broadly neutralizing antibody in infants, whereas on average, the broadly neutralizing antibodies that have been studied in adults have about 20% mutation. So we're excited that infants are doing this differently. We think we can learn from how they're doing it. We don't really understand if infants just do these things differently for all infections, and that's, I think, a big question that we probably won't be able to tackle, but it's interesting to think about from these studies. Okay, next slide. So this is one of the outcomes of the Infant Antibody Project. These are the 3 women I talked about who worked on this project, and you can see that they soon after had infants of their own, all 3 of them. Okay, next slide. So let me just close with a few minutes of telling you about the collaboration and some of the other things beyond the science. Next slide. So, you know, I started working with Ruth Ndwadi and Dorothy Mbori Nagacha when we were, well, I won't say how old we were, but we're all about the same age, but in the 90s. And, you know, you can see us here at a meeting in Nairobi in the early years. I've stayed close friends with them. There's a picture showing me with Ruth a couple years ago and her family shown there. Her son's so tall that his head's missing in the picture. And another part of the collaboration, Grace John Stewart, who was an ID fellow at the university at the time working in Kenya, spent 10 years of her life in Kenya, is now a professor and well-known leader in maternal child health at the University of Washington. And Ruth went on to be the chair of the Department of Pediatrics. And after Ruth, Dorothy went on to be the chair of pediatrics. And so they really have had an incredible impact in Kenya with the science and the education that they do. Next slide. The other thing that's been important in this large collaboration that's been, you know, long-standing is that it's bi-directional and it's also about training. So I've had 3 Ph.D. students from Kenya who came to my lab and got their Ph.D. from the University of Washington, went back to Kenya and really became important drivers in different aspects of science in Kenya, leading the way in efforts there. Next slide. One of the things we also think is important is, you know, capacity building. As I mentioned in the beginning, my lab basically was trying to help develop assays to diagnose HIV in infants, and we did that in my lab in Seattle. But obviously that's something we wanted to get on site, and all these assays that we have developed over the years, we've tried to transfer back to Kenya. Next slide. And we did that really aggressively in 2005 because one of the first PhD students in my lab was returning. So we had somebody who really could go back and lead the laboratory there. And we really felt there was a need for infrastructure and on-site work rather than it being done in Seattle. So we were fortunate, I guess, to get a lab space, and that's shown here, our first lab space that we got for virology at the University of Nairobi. But we bravely went ahead and made a lab out of this. Next slide. Next slide. And this is the lab at the University of Nairobi where we now do molecular virology, and this is led by Bhavna Chauhan, who was a PhD student here a number of years ago. Next slide. So I hope that this sort of gave you this picture of the kinds of ways that people can collaborate internationally and in different disciplines. Ruth, Dorothy, and Joan and Grace started out trying to understand the risk of breast milk transmission and did the Nairobi trial for that reason. Because of the work that they did, the samples they collected, the data, the collaboration, we've been able to understand unique aspects of the infant antibody response. And this has all also been part and parcel of training and capacity building as well. And actually a couple of the students from Kenya worked on aspects of the infant antibody response while they were in the lab. Next slide. So I'll show you just one more phylogenetic tree, and that is all the people who did this work in my lab, did the lab work, obviously in collaboration with the groups that I've told you about. And this is a phylogenetic tree that was commissioned as an art project, as an artist project for the 30th anniversary of my lab. And it shows, you know, how the lab started and how people radiated out and became progeny of the person before them in terms of the projects that we do. Next slide. And final slide is just to show that we have a big tradition of Halloween costumes in my lab. And this last year we were the baking show following the British baking show, and people did an amazing job making some pretty impressive desserts that day. So that's a picture of the current lab right there. So now I am very happy to take questions. I think we have some time of all natures, including what we do, as well as, you know, virology in general.
Great. Thank you so much. That was a wonderful overview of the path of your career, and it's very interesting to see how the collaboration has been sustained in different ways over time. So please feel free to unmute yourself. We're a smaller group, so if you wanted to just unmute yourself and ask a question, that's fine, or feel free to type into the chat. I told Julie how students and teachers in this forum have been asking a lot of questions, so.
I'm ready for them.
One of the things that I was kind of wondering about was you had mentioned early on that there's the period in the breast milk transmission studies where early on the infants are having higher mortality and not able to fight off the virus as well. And so I was wondering how that connects to their ability to make more, this polyclonal response. So are was the infant that you're looking at kind of past that initial point where, I mean, they were still alive, so that's right. An indicator, right? So, but what happens? Does it take a while for the immune system to just kind of get going for the infants, or what happens?
It does. You actually bring up a good point, and it's why I think nobody looked at infants at their antibody response before, because their disease is more progressive. And so it's counterintuitive if you think antibodies would protect, but yet these infants who don't do well have the better antibodies. And part of that conundrum, I think, is what protects from infection is probably different than what can control an infection that's already established. So I think that there's not good evidence that if you gave neutralizing antibodies to somebody who'd been infected for a long time, that they would do much good. They need to be there to block the initial infection. And so some of these infants, for example, one that we studied in detail kind of defies all odds because it was an infant infected in utero, born prematurely, but still was alive at the end of the study and had an impressive antibody response. But you don't have to have, you know, disease is not correlated with better antibodies, protection is. Looks like there's a question.
Yeah. What is the current suggestion for new moms who are HIV positive? Still formula?
Yeah, you know, so the WHO still recommends that women who do not have access to clean water breastfeed. Of course, now mothers and infants are both treated. So remember I mentioned that people are more infectious if they have high viral load. So if you treat mothers with antiretroviral therapy, you reduce their viral load, you reduce their infectiousness, and then you give the infants treatment as soon as they're born to protect them from infection. So the mortality and morbidity associated with lack of clean water and formula is significant. So WHO, for that reason, still recommends, if there's not access to clean water, that women breastfeed.
Thank you. What's happening with the study that you described now? What's some of your latest work that's related to this study?
To the antibody study, we're trying to, you know, when you just study one case, you never know if it's the odd case or that it's truly something infants do differently. So we're trying to study additional cases. There's a lot of interest because of this result, and then our result was reproduced in a South African cohort a couple years ago, that if infants really do something differently in general, whether if we get to the point where we have an HIV vaccine, we should be vaccinating infants rather than waiting till they're sexually active teenagers, which is what we do with the HPV vaccine. So we need to show that this is something that is a consistent response in infants. And we've really only dissected that one case very carefully. Just getting those antibodies took 2 years of brute force. So it's slow going to do multiple cases.
Okay, were you surprised that only one antibody was broadly neutralizing in the infant study?
Well, we were, but we kind of knew that there wasn't going to be like a heavy hitter broadly neutralizing antibody in the infants because we'd kind of mapped their responses in a different way before we launched into the big study. So I thought it was going to be a mixture of different antibodies, but I thought there might be several that were at least a little bit broad, but we also could have missed some. One of the things about this is that there's a lot more B cells in your body than what we sample with a couple mils of blood. So there's a lot we can't get to when we do these studies.
How long did it take to do the screen of all the B cells that you had available to you there?
It's a long process. So we do part of it with a robot because you have all these plates, 384 wells in the plate and 100 of them, and you're doing all these assays on very small microliter volumes. And then, you know, somebody says, well, these wells look interesting. It looks like maybe they're making neutralizing antibodies. You have to go reconstruct the antibody and test it. And about 2/3 of the time, you're actually wrong. So you make a lot of antibodies that aren't that interesting in order to get a few that are. And you'll hear about with SARS-CoV-2 that people are doing this exact same thing. They're trying to isolate these antibodies going through this similar process.
Good. Other questions? You know, one other thing that I really have noticed and appreciated in your work is your mentorship of so many young scientists, and you've been recognized for that and received awards for that. And have you found in general that that's been something that's also been supported by the administration, that, you know, you've been working with scientific administrators? Has there been pressure to, you know, not have so many folks that you're mentoring, or, you know, what are your thoughts about mentoring in general?
Yeah, I mean, that's a good question. You know, graduate students in particular, and most of the data I showed you was from PhD students. When they first come in the lab, and you probably remember this, Jeannie, because I know you were in Nancy's lab, that, you know, it takes them a while to get going. And there are some labs that take the view that if they can't get the project started and done in the right time frame, then they will move on and get it done another way because they want the first paper and they want the big, you know, splash. And you do have to kind of decide, are you going to think about each student learning and doing well, or are you going to be a big splash kind of lab and go for the competition and the first one to get there? And, you know, I think over the years I've taken the approach that the training is important, and in the end the students really do nice work. It just takes them a while, but there's a wide range of perspectives on that, and I've had fantastic students. And one thing about probably the approach I take is that people know about my lab, and, you know, it's like if you play at UConn, you know, they know about this team that does well, and so they want to join it. And I think in the end, you know, 30 years or more, it's worked, but it is a big challenge for faculty to make that decision because they're under so much pressure for the immediate kind of satisfaction of the big paper.
We do have a couple students on the call that I recognize. What would you say to students in high school who are just kind of starting out in their scientific careers? Is there any advice that you might give them about, you know, their focus and then pathways?
Yeah, I mean, I have to admit that as a high school student, I wasn't that interested in science. So it's funny that I've ended up being a scientist. But I think I think it's really important to see if it's your calling. And that's why these programs and being engaged now is important. And it doesn't mean it will be. It's okay if you say, I'm excited about science, and then you start doing it and say, you know, it takes too long to get an answer. This is not my personality. But I think if it is for you, the thing for me that's been great is not only with the students and training, but, you know, you get to follow your ideas. And I just don't know of many careers where you just get to say, I think you can do it. So, you know, that's just really exciting. And for example, when we sort of decided to do the infant antibody project, it was, I think I rode my bike into work that day and I was thinking, you know, there's something about infants that's different. Maybe we should just screen them. And you just get to do that stuff. And I don't know of many things like that. So it's an exciting career, but you do have to be somebody who will wait for the long game because, you know, it takes a long time. Sometimes things fail and fail. So I guess those would be some of my thoughts.
All right. Thank you so much again for joining us today. And thanks also to all of you who came, and wanted to let you know next week, Julie Hill from SciTech will talk about vaccines, technology, and public health. And she actually is a specialist in a process called flow cytometry. So she'll be talking a lot about technology and how technology and research are intertwined. And we'll continue to kind of think about how, especially in the world of HIV vaccines, there's an application to the tools of flow cytometry. So there'll be a thread that continues on there. We're decided to go through the summer at least, and probably into the fall with Hutch Home. And I hope you, you know, continue to spread the word about it. We'll take a break on July 7th, but all the sessions are recorded and posted on SEP's event page. If you miss a talk and you want to go back and catch it later, I also know a few teachers who are sharing them with their students, so feel free to do that. And I pasted the evaluation feedback form into the chat, and you can let us know if you have thoughts about any of our sessions. And can you unmute yourselves and give Dr. Overbaugh a round of applause? And you can also enter in your appreciation into the chat.